Okay, so welcome to this next video in which we are discussing the adaptive immune system and the immunosuppressant drugs. Okay, so we're just discussing how you synthesize guanosine monophosphate. So, we've discussed that you start with inosine monophosphate, and then the enzyme inosine monophosphate dehydrogenase converts this into xanthosine monophosphate, ZMP. ZMP is then converted into guanosine monophosphate by the enzyme GMP, or guanosine monophosphate synthase. Okay, so let's discuss how this reaction actually occurs. So basically, what you're going to do is you're going to bring in an ammonia molecule. Okay, so on this side of the reaction, okay, so we're looking at this conversion here. So you're going to bring in an ammonia molecule here, which is a nitrogen linked to 3 hydrogens. Okay, you're also going to bring in a molecule of adenosine triphosphate, ATP. Now, on this side, what you will have is the guanosine monophosphate, and you will also have an adenosine monophosphate plus a pyrophosphate molecule. Okay, so what you have done is you have chopped off the 2 terminal phosphate groups on this adenosine triphosphate molecule, and those have come off as this pyrophosphate molecule here, and you've then got the adenosine monophosphate here. So let me just draw a little cartoon for this. So if this represents our ribose within our adenosine monophosphate, here's the organic base adenine, and by the way, adenosine means adenine bound to ribose, just as guanosine means guanine bound to ribose. And then you'd have your 5th carbon up here, and then off that 5th carbon you would have 3 phosphate groups, 1, 2, 3. What you're going to do is chop off 2 of them. You're going to chop here. That will chop off 2 phosphate groups that will remain bound together, which is what's known as pyrophosphate here, PPI for short. This is pyrophosphate. And the remainder, which is adenosine now with a single phosphate on, is called AMP. Okay, just as guanosine with a single phosphate is called guanosine monophosphate, GMP. Okay, so to chop those 2 off, you need to hydrolyze that bond. You need water, basically, to hydrolyze that bond. So the water must come from this conversion, basically, that's going to chop the ATP into the AMP plus the pyrophosphate. Okay, so let's see how we're going to do this. So again, this isn't a molecular mechanism, this is just helping you to keep track of everything. So imagine splitting this double bond between the oxygen and the carbon. So there are 4 bonds overall, there are 4 electrons overall in those 2 bonds. 2 from the oxygen, 2 from the carbon. Give the 2 from the oxygen back to the oxygen, and the 2 from the carbon back to the carbon. Okay, then cleave this bond here. Again, there's an electron from the hydrogen and an electron from the nitrogen. Give the electron from the hydrogen back to the hydrogen, and the electron from the nitrogen back to the nitrogen. Then reforge a double bond here, basically. Make another bond between the nitrogen and the carbon alongside the existing one. So the nitrogen now has this free electron here. The carbon now has 2 free electrons. It will use one of its free electrons to form another bond with this nitrogen, making the double bond here. Okay, then take this ammonia molecule here, which is nitrogen linked to 3 hydrogens, like so, and break one of the bonds between the nitrogen and the hydrogen. Okay, here. So cut off one of these hydrogens. Give one of the electrons back to the hydrogen and one back to the nitrogen. So the nitrogen and the hydrogen both have a free electron. Then bind this nitrogen to that carbon's final free electron. So remember, it had 2 free electrons. One it's used to make this second bond with this nitrogen, and the other it's going to use to make the bond with this amine group, which will put that on. So now we've got guanine, okay, and we should have produced water somewhere, which is going to hydrolyze the ATP into AMP and pyrophosphate. We've got our oxygen here with 2 free electrons, and we've got this hydrogen and this hydrogen, each with 1 free electron, so we can therefore form water temporarily, but then of course the water is involved in the hydrolysis of the ATP into the AMP plus pyrophosphate. And the reality is you won't form water in the middle. It will go straight into the hydrolysis of the ATP into AMP plus pyrophosphate. Okay. But to help you keep track of everything that's going on, that's how this reaction as a whole makes sense. So what's on this side does add up to what's on this side, basically. That's the intuition for this. Okay, so that's how this enzyme, guanosine monophosphate synthase, synthesizes guanosine monophosphate from the xanthosine monophosphate. So if you give the drug mycophenolate, it will block this inosine monophosphate dehydrogenase, which is going to stop the synthesis of guanosine monophosphate. Okay, now guanosine monophosphate is needed to make guanosine triphosphate, which is what you use to build RNA. Okay, so let me just explain this. Let me get another piece of paper. Okay, so when you're building a strand of RNA, okay, so let's say we've opened up the DNA. So here is our DNA that's now opened up. Okay, and we want to build a strand of mRNA that's complementary to the coding strand of the DNA. So let's say this is the coding strand of the DNA, and this is the non-coding strand of the DNA. So I'll colour that in a different colour. So in blue here, the dark colours like blue and turquoise. Those are the DNA. This is the coding and this is the non-coding strand. And here we'll have in a very bright colour is our mRNA. Okay, now let's say you want to add on another nucleotide onto this mRNA. And let's say your organic base over here on the coding strand of the DNA is a cytosine. Then you need to add on a guanine next along. And to do that, what you do is you bring in a guanosine triphosphate molecule, and you're only actually going to add on the guanosine monophosphate, but what will happen is the pyrophosphate, the two final phosphate groups, will fall off basically. And the energy that's released from this reaction will drive the addition of the guanosine monophosphate nucleotide onto the mRNA. So, if you do not produce any GMP, guanosine monophosphate, you can't produce GTP, because GTP is made by just adding two phosphate groups onto the guanosine monophosphate. Okay, so if you can't produce any GTP, then you can't make mRNA, because you're going to come across cytosines, basically, within the coding strand. It's roughly a quarter of all the organic bases that you're going to have to add on to this mRNA are going to be guanines. Okay, so if you can't make guanosine monophosphate, then you're not going to be able to make mRNA. So protein synthesis is going to stop basically, because if you can't make mRNA, then you can't make protein, because mRNA is needed to make the protein. So protein synthesis is going to stop, or at least go down. Now, also, guanosine monophosphate is used to make the DNA nucleotide. Okay, so remember the difference between the nucleotides in DNA and the nucleotides in RNA is that in RNA they are ribonucleotides, so the sugar molecule is ribose. Okay, whereas in DNA it's deoxyribose. And the difference between the two is, if I bring back my picture, this is ribose. Deoxyribose would not have this alcohol group here. Instead it would have a hydrogen off that carbon. Okay, so you need to create DNA or nucleotides as well, but they're created from the guanosine monophosphate. Okay, so guanosine monophosphate is the nucleotide that is in mRNA, or all RNA, but the nucleotide which is in DNA is what's known as deoxyguanosine monophosphate. Okay, so that's denotes the deoxyribose that you have. Okay, so guanosine means guanine plus ribose. It does not mean guanine plus deoxyribose. So instead, deoxyguanosine means guanine plus deoxyribose. Okay, so let me put this down here. So deoxyguanosine means guanine plus deoxyribose. Okay, so in order to make DNA, you're going to need deoxyguanosine monophosphate. Okay, and this is made from the guanosine monophosphate. So if you have no guanosine monophosphate, you're also not going to have any deoxyguanosine monophosphate. Okay, and if you don't have any deoxyguanosine monophosphate, you can't copy the DNA basically, because that's what you need in order to add into the DNA. Well, again, you're going to add in deoxyguanosine triphosphate, but of course you synthesize deoxyguanosine triphosphate from deoxyguanosine monophosphate. Okay, so also DNA replication is going to be hugely reduced if you can't produce guanosine monophosphate. So remember, the two essential things if you're going to divide from being one cell to being two cells are that you need protein synthesis to copy all of the essential proteins, and you also need to be able to replicate your DNA so that you can replicate the genome, so that each new cell will have a copy of the genome. Okay, so mycophenolate is going to stop protein synthesis and it's going to stop DNA replication. Now, that will stop cell division of these T and B lymphocytes. The question is, why doesn't this have such a strong effect in all cells? Why is it selective, or at least in part selective, for T and B lymphocytes? Well, the reason is that other cells have other ways of synthesizing guanosine monophosphate other than this, whereas these cells, T and B lymphocytes, lack these other ways of creating guanosine monophosphate. So when you inhibit the inosine monophosphate dehydrogenase, you completely cut off all synthesis of guanine nucleotides, which stops you being able to produce mRNA and also stops you being able to produce DNA, stopping DNA replication and protein synthesis, and therefore preventing cell division within the T and B lymphocytes, and therefore having a very powerful suppression effect on the adaptive immune system.